So I'd like to introduce our fourth session, which is going to be about robustness and geometry in biological systems. So our first speaker is Stanislav Shvartsman, who is a professor at Princeton University. And he'll be talking about a class of natural tessellation algorithms. So Stanislav Shvartsman is professor of molecular biology at the Louis Siegler Institute for Integrative Genomics at Princeton University. The Shvartsman Lab develops and tests mathematical and computational models for how cellular systems self-organize into functional structures during development. So with all of that, I'll let Stanislav Shvartsman get started with his talk.
Thank you very much for the introduction. Not being a neuroscientist, I'm definitely learning a lot from every talk. I will tell you about the work that is done primarily by three people in my lab. So a former graduate student, Audrey Zhu, a postdoc, Liu Yang, and Harrison Oatman, and I'll acknowledge other collaborators. So another canonical example of self-organization is synchronization of oscillators. So it started with an observation in the 17th century by Christian Huygens, who noticed that the two pendulum clocks that are on the same wall tend to synchronize and go in phase. So the legend goes that he was sick in bed. He noticed this, and he modeled it first in a physical experiment where he took he didn't work with the clocks. So he took two simple mechanical penduli, placed them on a common support, and observed synchronization as well. So he already knew used Newtonian mechanics, and he came up with a correct explanation for synchronization that has to do with transmission of forces through the common support. So this is actually a common phenomenon. Synchronization of oscillators is observed not only in mechanical systems. You can observe, let's say, at the time of Hanukkah, the synchronization of candles that are burning nearby, and there the coupling through stealing of oxygen from each other and generating convective flows that synchronize the candles. Synchronization can also be observed at the level of organisms when swarms of fireflies start to flash in sync, and there it is not just an accidental pattern, but is believed to be important for mate selection and recognition of conspecifics. So study of these systems is honeypot for physicists and applied mathematicians. It is a very, very active research field. There are several books written on the subject, and the books range from relatively popular books like this wonderful book by Steve Strogatz to technical books written by Arkady Pikovsky and colleagues. If you study this literature, then we'll realize that most of the work has been on systems where the number of oscillators is constant. So you have three candles that are placed in a triangle. They synchronize. They can form different patterns. You have several penduli that are coupled on a common support, 2, 3, 6, and they synchronize. So I will tell you today about a class of developmental problems that also have to do with synchronization of oscillators, but they have to do with dynamical systems where the number of oscillators is constantly changing as a function of time. And I will try to argue that this is an absolutely natural context for what is happening in development. But first, what oscillators are we talking about? So we'll be talking about mitotic oscillators. So if you study mitotic cell cycle in high school, you will learn about the gap phases, but the gap phases are there just to make sure that all the conditions are met. The real objective is that you need to first replicate genetic material and then separate it into two daughter cells. These activities should be under no circumstances overlapping in time. To make sure that they are not overlapping in time, there is a chemical clock that tells the cell that now they should be replicating DNA, replicating genetic material, and then having replicated and having checked the correction of the replication, you separate it into two daughter cells. So there is a chemical rhythm that corresponds to the differences in the activity of a certain protein kinase or a class of protein kinases, cyclin-dependent kinases, that tell whether the cell should be replicating genetic material or separating into two daughter cells. So what is this rhythm? A mathematical metaphor for this rhythm is a limit cycle oscillator. So this is the simplest possible form of the limit cycle oscillator. So here we have a phase that moves constantly increasing, just like you would have in a perfectly harmonic oscillator. And then we have a nonlinear equation for the amplitude. And the nonlinearity is, so first there is a linear term that says that when the amplitude is low, the system will self-excite. And then there is a nonlinear term that kind of reins in the amplitudes when it becomes too large. As a consequence, in contrast to what is happening in a harmonic oscillator, such a system always converges to exactly the same amplitude, as opposed to what is happening in a harmonic oscillator where everything depends on the initial conditions. And this convergence is a consequence of A, nonlinear dissipation, and B, important functionally because you can rely on the same amplitude and on the same frequency. All right, so this is a mathematical metaphor. In reality, what is happening is that there is a quite complicated circuit of biochemical reactions that involve pretty much every chapter from the textbook of cell biology. There is synthesis of cyclin, which is an obligate partner of a cyclin-dependent kinase without which there is no activity. In addition to the formation of this complex, the different parts of the complex have to be phosphorylated in order to either increase or decrease the activity of cyclin-dependent kinases. And phosphorylation depends on kinases and phosphatases that in turn control the activity of this kinase. On top of everything else, there is a degradation of the cyclin that makes sure that the system generates a self-sustaining rhythm. So this diagram, however complex it is already a grossly simplified version of what is happening, even in the simplest versions of such mitotic oscillators, which are actually observed in early embryos, such as sea stars and sea urchins and frogs, where the essential components of the cell cycle have been discovered. One way or the other, through the beautiful work of people like Jim Farrell and John Tyson, it was established that in early embryos, there is indeed a periodic orbit of activity of cyclin-dependent kinase, cyclin complex, that comes through the collective activity of positive and negative feedback loops that act as in correspondence to different terms in the mathematical metaphor that I have just mentioned. So something like this, modulated by the gap phases that make sure that the cell is of appropriate size, that all the chromosomes are attached properly, that there is no DNA damage, operates in every mitotic cell cycle. However, there is a special type of cell cycles that I will be describing today that do not involve any cell growth. And they have to do with something that is called reductive cell divisions, which happens in the development of every animal. And the animal can be a mammal, such as ourselves, or it can be a fruit fly, such as one of our favorite model organisms, and certainly the main model organism in our lab. So when you start your development as an animal, you start as a single cell. The single cell is a fertilized egg. Eggs are absolutely remarkable cells. So in every single organism, this is the largest cell by volume. There are cells that are certainly very long. So these are the neurons. But oocytes are the largest cells by volume. And they have to be so large because there is a part of embryonic development, the length of which varies depending on which species you are, when you must accomplish a lot of different things without any help. So for example, if you're an insect, the egg is deposited, the fertilized egg is deposited in the environment, and there is no addition of matter. So there is oxygen that comes in that allows the embryo to breathe in order to get the ATP, but there is no addition of sugar or protein or fats. So the fertilized egg must complete embryogenesis and give rise to larvae with nervous system, complicated behaviors, muscle responses completely on its own. You would say that mammals are different because there is placenta and maternal-fetal interaction. However, it turns out that by the time that the embryo implants and just starts interacting with the mother and receiving products through the blood supply in the placenta, the embryo already accomplishes quite a lot. So it goes through cleavage divisions where the cell is first partitioned into 2, and then into 4, and then into 8. So by the time that the mouse embryo implants, it already has formed 128 cells. These 128 cells have been assigned to 3 different cell types. All of this happens as a consequence of cleavage divisions, where one cell is partitioned into progressively larger number of cells. It's these divisions that I will be talking about. Because every division reflects a turn in the crank of a mitotic oscillator, every time this happens, you give another oscillator to the system. Okay, so these are the two systems that I chose as examples, not because we work on them. So on the fly in my own lab, on the mouse in collaboration with my colleague Esther Poshfai, but these systems could not be more different from each other from the standpoint of self-organization and pre-patterning. So the fly egg is 0.5 millimeter long. The mammalian egg is 50 microns in diameter. The mammalian egg has absolutely no information. So it's a completely symmetric system. The sperm can enter at any point. There are no maternal products that are localized. Whatever the system does, it does on its own. It breaks symmetries. It partitions cells to different types. It makes sure that these types are apportioned to different categories. So this is an example of a purely self-organizing system. The fly is completely different. So the egg is much larger than the mammalian egg. And the reason for this is easy to understand. Mammalian egg eventually will implant and start giving rise to the fetus. The insect egg has to do everything on its own until it forms an animal that can move and fend for itself and behave. Okay? So the egg is large. In 2 hours, it goes through 13 sequential divisions. So this is absolutely blinding speed. So this is probably the fastest cell cycle known to man, woman, or child. Okay? So in 2 hours, it forms, so if you go through 13 divisions, so 2 to the 10th is 10 to the 24th, 2 to the 11th is 2,000, and so on and so forth. So 2 to the 13th is close to 8,000. You don't form 8,000 cells, but you form approximately 6,000. And these 6,000 cells, by the end of the 2nd hour of development, are distributed uniformly under the common plasma membrane. A very small group, and these cells are precursors of every somatic part of the future animal. An extremely small part, minor part of these cells are the so-called pole cells. So these cells have to be moved to they go outside of the embryo, and these are the precursors of the future egg and sperm cells. So by the end of the 2nd hour of development, the system has formed a very, very large number of cells by mammalian standards. Furthermore, it has already started to read out the broken symmetries that exist in the system as a consequence of symmetry breaking and pre-patterning events that happened during the egg development. The symmetry breaking is of 2 different flavors. First, there are localized transcripts such as the mRNA forbicoid that Fena has already mentioned. Then there is a mixture of RNA and proteins that give rise to the future germplasm that are necessary to form the future pole cells. And then there are 2 types of modification of the extracellular matrix. There are red modifications that are present at both poles of the embryo, and there is a blue modification that is present at the bottom of the embryo. So during the 45 minutes into the 3rd hour, having formed this uniform field of cells, having assigned approximately 30 cells to the precursors of the future germline, the system patterns itself and gives rise to patterns that look like the beautiful stripes that you read about in every textbook of developmental biology. So the patterns look like anterior-posterior stripes that exist pre-existing symmetry, broken symmetries in the AP direction. They look like bands that are non-uniform in the dorsal ventral direction that reflect modifications of the extracellular matrix on the ventral side of the embryo. And then there are modifications at the poles of the embryo. So the beautiful part about the system is that it allows to study it using a combination of genetics, imaging, and biochemistry, because the system is sufficiently large for us to measure the composition of the system. Okay, so I will try to illustrate how in the last couple of years we started to combine genetics, biochemistry, and optogenetics with some mathematical metaphors for self-organizing systems to understand the interplay between pre-patterning and self-organization of the system. And before I go any further, I want to illustrate that this strategy is rather different from what is happening during mammalian pre-implantation development. The mouse embryo forms cells and patterns them at the same time, assigning them to the precursors of the future placenta, primitive endoderm, and the epiblast. This happens at the same time. this system, because speed is so important, decides to first expand the primordium and then pattern it. So the patterning is not self-organized, but the formation of this uniform sheet of cells, the formation of something uniform, is in fact self-organized, as I will try to illustrate when I show you the results. Okay, so these are the things that can be done in this system. Okay, so what you see on the left is a data set from a light sheet microscopy experiment where we take, the movie lasts about an hour, where we take an embryo that contains GFP tagged histones. Because we have GFP, we can see nuclei. And because of the advances of microscopy and computer vision algorithms, we can not only see nuclei, but see every single nucleus, and we can count them. Okay? So we can see the doubling of the number of nuclei from nuclear cycle 10, 11, 12, 13, and 14, right? So we can observe how nuclei are forming in the system, and we can study the formation of different mitotic domains, and we can also study macroscopic properties such as density. Okay. So imagine we take a group of nuclei somewhere here, and we want to do simple clonal analysis. We want to look at all of their progeny, which is bread and butter of a lot of experiments in developmental biology, except that here we do it by imaging. There is no need to label anything because we have decent image processing. So if we take a group of 8 nuclei, we are going to, and so the time here is in minutes. So just see how much is happening in 50 minutes, okay? So we see that we go from 8 cells to 16, to 32, to 64, to 128 with just remarkable synchrony. If we take any other group, localized group, we are going to observe exactly the same synchrony. So the divisions will be synchronous, and not a single nucleus will skip a beat, will skip a step. A very, very small fraction, less than %, will not do this because the nuclei will fall out in the yolk, but this is really a minor fraction. So divisions are in a syncytium. So at this point, because the system wants to go very, very rapidly, it is still, nuclei divide, but the system still has not built the membrane. So the entire system is one large cell where things are coupled by diffusion in the cytoplasm. So these divisions are synchronous, and you can say it's not surprising because we're sitting in the common cytoplasm. But just as in medicine, we realize that we have a certain part of the body when it hurts. In this case, you realize that this synchrony should not be taken for granted because you see an example of how easily this synchrony can be lost. So as I told you, there is a group of nuclei that leave the syncytium, they form membranes, and they start forming precursors of the future pole cells. So it turns out that if we do the same, yes?
Can I just ask a clarification?
As many as you want.
So what is the rough timing here is 10 minutes between divisions?
10 minutes between divisions, and then it gets longer and longer.
And what is the diffusion time scale?
The diffusion time scale is actually quite short. So you can, we can do the math.
Okay, so it's like it's a well-mixed system.
No, it's not a well-mixed system because otherwise you would not form gradients. So it's L squared divided by D. The L square it's 100 microns. So it's 10 to the 4th in the numerator. The diffusion, even if you are very generous, is 1 square micron per second. So if you divide, you need more than an hour to go through the entire egg. So it's eggs and oocytes are very special cells that cannot rely purely on diffusion. And you will see that in the system, it relies on diffusion only locally. But in addition to diffusion, it relies on positive feedback that generates propagating waves that are very similar to the morphogenetic furrow that Robin has already mentioned in his talk about the eye. Okay, so this was a great question. Okay, so if we follow, so the main point so far is that synchrony in the system should not be taken for granted because if we take nuclei that have exactly the same genome, exactly the same age, they lose synchrony. They do not divide synchronously, and some nuclei just skip a division. So what is the origin of this synchrony? The origin, the key to answering this question came from the fact that, so I'll play this movie several times, that somatic divisions happen in waves, So this was known for a long time, and this has been studied since 1940s, but using techniques that are extremely painstaking. So what you are seeing here is that a movie in which we taught a computer to recognize a nucleus that divides, and this nucleus is yellow, from a nucleus that is in the interface. So what you can see is that divisions start from the poles and they propagate inside. Okay, so there is a propagating wave of activity. These waves are not of constant speed, but there are definitely a propagation, very similar to the coherent events that were mentioned in the talk of Matthias and his collaborator. Okay, so we have an oscillatory system where every nucleus carries a mixture of chemicals that allows it to go around the cycle to make more and more nuclei. And what we see is that in these systems, events start from a certain parts of the embryo. So where else in biology do we see propagating waves? Okay, so one example is a pacemaker in the heart. So what is happening in the heart? So we have propagating waves of electrical conduction that generate mechanical contractility. And these waves always initiate under normal conditions. They initiate from the same part, which is called the sinoatrial region, which is the local anatomically defined and transcriptionally defined pacemaker that has a higher frequency. So if we have an oscillatory medium and we place in it a defect or a group of oscillators that oscillate at a higher frequency, it will entrain the rest of the medium. The system will be oscillating, but the oscillations will be always starting from the same place. Okay, so what I will show you now in just a couple of steps, that we discovered a true pacemaker in the system. We understood, so we know that waves initiate from the poles. So we decided to look at the poles, at the source of something that can speed up the mitotic cell cycle. So first of all, are the poles different biochemically? Yes, they're different biochemically, and this was known long before us as a result of the giants on whose shoulders we stand, like Trudy Schubach and Christian Lusten-Wolhard. Okay. So it was long known that at the poles of the embryo, there is a locally activated kinase, which is called an extracellular signal regulated kinase that is very important in the context of mitogenic signaling. And this kinase was important for transcriptional patterning of the embryo. So what we discovered is that the same kinase also plays a role in speeding up locally the mitotic oscillator that runs in the system. If this is true, then we should be able to have disorganized oscillations when we get rid of this local pacemaker. And second, we should be able to generate an additional pacemaker if we activate the system in place where it is not normally active. So first, let me describe a second experiment. So in this experiment, we placed this red enzyme that is present everywhere but is active only at the poles of the embryo under optogenetic control. And we pointed the laser that uses blue light to activate the system only in the center of the embryo. So it doesn't matter how it happens, but this is a robust way to activate the same system by light. So what you see here does not even require a voiceover. Okay? So we start a wave from the middle of the embryo. So this is completely reproducible. So it means that we can watch the system, we can use machine vision to process the patterns, understand that patterns are initiated, that waves are initiated at the poles, have a hypothesis, shine the light in the center of the system, and completely change the pattern. Okay. So if you have pacemakers in a medium that is not as beautifully organized as the example that I showed before, but is actually operating in a chaotic regime, you can use a pacemaker to entrain the system and convert it from a disorganized spatiotemporally chaotic regime to the one that has an organized pattern. So if this is the case, that one should expect that if one removes signaling from the poles of the embryo, the oscillations that we have in a system should become disorganized. Okay. So this is what we have done. This is the normal pattern. This is the pattern that we ablated using relatively simple RNA experiment. The top is a control that uses cyclin E, which is the cells that are in the S phase. And what you see in the bottom are embryos when there are no pacemakers in the embryo. I hope you can see that the top is essentially synchronous, and this one becomes asynchronous relatively quickly before it dies a horrible death. Okay? Now, so we have a model for a pacemaker for mitotic activity, but this, the problem with protein kinases is that when you phosphorylate glucose, you phosphorylate only this specific position in a glucose. When you phosphorylate a protein, you phosphorylate this specific serine or this specific threonine. So these kinases can have tens or hundreds of substrates in any given context. So how exactly does this red kinase feed into the mitotic oscillator? So we had a fantastic collaborator with Martin Wurr, who is my neighbor and friend, both in the township and in the Genomics Institute. And we have done an experiment where we have stimulated the embryos by a relatively short 10-minute pulse of the blue light, generating a relatively uniform high level of signaling. And we compared phosphoproteomes after 10 minutes of stimulation. We found a whole bunch of proteins that are not important for this discussion, but the most remarkable thing is that we discovered phosphorylation sites on the core components of the mitotic oscillator, okay? Is it just something that is phosphorylated, or is it something that is also important? It turned out that these sites are important because we then made an experiment where we took candidate sites in the components of the core mitotic phosphorylation, converted them into alanines such that they cannot be phosphorylated, placed them in the posterior of the embryo, and shown that when this happens, the top is a control, and we see that the posterior is immediately becoming different, and again, this embryo dies. Okay? So what we established is not only that there is a pacemaker, not only that we can generate an ectopic pacemaker, not only that the system loses synchrony without this pacemaker, but we understood at the level of specific amino acids of a specific substrate of the enzyme that is activated in such a pacemaker fashion. So now I want to make the connection to the title of the talk that actually talks about tessellations. Okay, so let's watch again the movie that describes how cleavages are happening in the common cytoplasm. So two things are happening. So far, I focused only on the fact that there is more nuclei in the system. But you also see that there is a lot of sloshing around. And you have all seen how mitosis is happening. It is actually a mechanical event. Cytoplasm is moved. There is cytoskeletal networks are remodeled. So there is plenty of activity. So let us start and stop and think what can be actually happening when you have a group of nuclei, let's say this blue, this cyan, and this gray, and they divide. Okay. As they divide in the common cytoplasm, they can be just cleaving the domain, just like you have a chocolate bar that you partition into different parts. They can also mix, because after all, it's a common cytoplasm, and there can be some intermixing. Or they can be deformed. Okay. So we can detect not only every single nucleus, but because we can both segment the nuclei and track them, we can also identify the clones of cells, and we can watch how the initial pattern of cells that is uniform is deformed. So this is actually data. This is the real embryo where we have detected every single nucleus by segmentation, where the lineage of every single nucleus was detected by tracking. And we see these jigsaw puzzles. And what we see, what we can conclude from these movies, and we have several embryos, is that these clones are deformed. Oops. These clones are deformed but not mixed, or shaken but not stirred. Okay? Okay. So this is the work that was done by Liu, Audrey, and Harrison. So Liu was the one responsible for all the genetics and optogenetics and material for phosphoproteomics. Audrey was a joint student with Martin, and Harrison established the vision algorithms. So why is this interesting? I'm done. So why is this interesting beyond this specific system? So this is just one insect. So if you look at many different insects, including cockroaches, okay, you will find that not all of them have uniform patterns of nuclear density. In some insects, there are parts of the blastoderm, which is arrangement of nuclei under the common plasma membrane where the density is not uniform. So thinking how one can predictably control the density of nuclei, which reflects the spatiotemporal pattern of nuclear divisions, is an interesting evolutionary question. I think this is also interesting beyond biology, because if you think about what the egg is accomplishing, it is actually a process of tessellation. You have a domain, so in the case of something like a frog or a fish, it's a tessellation in 3 dimensions. You take a sphere, you divide it into 2 hemispheres, and so on and so forth. In systems where cleavages are happening under the common plasma membrane, tessellation is happening in 2 dimensions. And tessellation is very common in art. So this is an example of a mosaic from the Moorish Spain. So this is 12th century. This is M.C. Escher's tessellations. Anyone who has ever solved the PDEs knows how important meshes are. So all of these algorithms, especially for irregular domains, which are important in, to construct chairs or airplanes, so these domains need to be meshed. So it's very interesting to think how one can systematically generate these meshes. But beyond meshes, these are examples of tessellations in engineering. So this is really an engineering marvel, which is a tessellated surface that allows an insect to see, and which is maybe a millimeter in length, right? And this is on a completely different scale. This is the Webb telescope, which is tens of meters in diameter, which is also tessellated and bent in certain regions that allows it to collect information about our universe. So I think that by thinking how natural systems solve tessellation problems when they design sensory systems like Robin mentioned in the talk, or when they cleave their eggs, we can get ideas for man-made devices. Thank you.
Yeah, so thanks for the excellent talk. And yeah, I have 2 microphones going around. So are there any questions?
Fascinating talk. Just 2 comments. One is actually a novice question. Isn't oscillation defined as something that comes back to its original state? In which case, are the cells really coming back to their original state, or they're actually at a different state now?
Okay, so so in the sense of the fact that, in some sense, the answer is definitely yes. It's the same original state if you think about the replication of the genome. So at some point, the genome has been replicated, the activity of DNA polymerase is ceased, and then it starts again. So from the standpoint of what you accomplish biosynthetically, the answer is yes. However, this is happening inside the cell. So DNA replication is only part of the things that are happening. If you think about the state of the cytoplasm, the state of the cytoplasm is actually drifting very slowly. And if you notice, these oscillations are becoming longer and longer. And the lengthening of these oscillations is absolutely important, because eventually the system will start caring not only about making more genomes, but about reading these genomes. So what I forgot to mention, especially in the context of all transcriptional profiling talks, is that all of this is happening before a single transcription event happened in the system. So it's, the system is really on its own. So from the standpoint of the state of the cytoplasm, it's not coming back to itself, it's slowly drifting. It's like the candle where the fuel is slowly being depleted, and as a consequence, it starts flickering longer. But from the standpoint of biosynthesis of genetic material, it's truly coming back to the original state. Yeah.
Related question to that. So the tessellation pattern, is it different, and it should be different on the edge versus what's in the center, right? Because it's got only one surface. So is that something to do with the overall shape?
Okay, good. It doesn't have so it is a surface of the egg. So it is truly, have you ever, I mean, do you eat eggs?
Yes.
Okay, so this, I don't know. Okay, you might be a vegan. Okay, but you know how an egg looks like. Okay, so it is happening on the surface of an egg that is actually more close to a shape to a banana. So it's an edgeless system. But what is actually important is that we notice that and this is the main point of the paper that Harrison is writing now, is that as the system is dividing, it is also helping itself to spread nuclei to make sure that the pattern is uniform. Yeah.
Okay, thank you.
So no edges, and that's an important thing.
Yeah.
Yeah.
So I also have two questions about the tessellation pattern. First of all, you said there's no mixing.
Mm-hmm.
But this maybe is just due to the fact that you only looked at a few cycles of the division. So these patterns became more and more complex, right?
No. So at some point, as you know from working with Eric, let me just first address this. So you know that eventually the system stops dividing at the 13. So we track them until mitotic division ceased. So the system kind of gets to the point of its bar mitzvah, stops dividing, and then it starts patterning. So at this point, there is no mixing.
There's no mixing, okay. But have you looked at the pattern of these patches?
Yeah.
Are they random, or is there any like?
It is random.
Is it?
Yeah.
There's no. And have you compared different flies? Do they show the same pattern? Or
Okay, so right now we have, I think, 7 embryos where we have done this. There are no systematic differences in embryos. The only difference is in the initial condition at the state from which the system starts cleaving. So you start from 1, and then you go to 2. And then when you go to 2 to the 9th, this is what becomes the starting point of divisions at the surface. So by the time they get to the surface, it is substantially, it is not really 512, but it is anywhere between 3 to 400. So this initial condition varies, which has to do with a different phase of nuclear spreading that has been really beautifully dissected by Stefano d'Italia, whom you might remember from your Princeton days. But once they're on the surface, it is just cleavages.
Yeah, I just wonder whether you can basically distinguish the model that it's just basically randomness from some local rules that govern this process.
This is precisely what we want to do now.
Yeah.
We have these movies where every single nucleus was tracked, and the plan is to try all the techniques that exist today in order to infer the rules in which this pattern is forming. The difference from more conventional, Kuramoto and oscillatory medium systems is that the number of degrees of freedom is changing. So this is non-trivial part where we have to develop some additional tools. But so far as I can tell, it's just local rules that generate the system. They just somehow push each other so that they deform. And deformation has to do, as you can guess, has to do with the way in which you place your spindle. Yeah.
Maybe in the same direction. First of all, this is a lot of fun, so thanks for that. The tessellation pattern is cool, but does it matter? I mean,
in this case, it does not matter.
Right? So it seems to me that the wave and the utilization of oscillation is the system's way of getting as close to synchrony as possible.
Absolutely. So the whole point is that the system wants to generate a uniform tessellation.
Right.
The precise...
Well, it doesn't really want to create a tessellation. It just wants to divide as synchronously as possible.
So,
the tessellation is
in
more like
what,
a side effect of
in what sense
not having enough time to just move away. I mean, it's deformed and not mixed, mostly because
so it is
if you so quickly...
Okay, it is tessellation in the sense that the clones are not mixed. This we did not.
No, it is tessellation.
Okay,
it's just that.
So we did know this in advance. What it wants to do is to establish a spatially uniform pattern of nuclear density on which you can then play out the program of zygotic transcription that starts from bicoid and dorsal and terminal patterning and it leads all the way to segment polarity genes and ushers you into gastrulation.
But the clonal heritage also becomes irrelevant, right?
We think, so my guess is that it is irrelevant. But there might be surprises because as you are patterning the system, the clones are moving. As a consequence, you might be exposed to a different level of nuclear dorsal or a different level of nuclear bicoid. And the folks who have been studying this for ever since the time immemorial are still arguing how exactly the transcription is read out and what is important for transcription. And the same question that Fenna discussed in the morning that has to do with spatial patterning of transcription factors, opening of the chromatin that has to do with the pioneer factor. They are as relevant in the system as they are in patterning of the mammalian brain. So I think that by being moved as a consequence of clones pushing each other in and out, you can be exposed to different environments. So I would not be surprised if the boundary cases might be pushed around into different phases. Yes, I will. Yeah. Yeah.
Thank you for the talk. I wonder when you optogenetically activate ERK, does that change the position of the pole cell?
Okay. So does it change the position
could
of
you
the pole?
repeat?
Yeah. Does it change the position of the pole cells? Okay. So first of all, the pole cells are evicted from the embryo before these cleavage cycles happen. So the pole cells already become the proper cellularized cells, and they're removed from there. Furthermore, we know that ectopic activation of ERK signaling in the pole cells because we can shine there. So under normal conditions, the embryo, and this is the work from Liz Gevis's lab and also from Ruth Lehman's lab, has shown that the system goes to great lengths to ubiquitinate and subsequently degrade the receptor tyrosine kinase that activates, that might activate ERK at the pole cells. But we know that when we activate ERK in the pole cells, nothing is happening. So the system can withstand it. Yeah. Yeah.
Right. So I guess one question was a follow-up from Robin's question, which is what would be the null model for your tessellation finding, which is that, you know, if you have cells that are just cleaving locally and then they remain kind of close by, then presumably maybe that's already it's a tessellating solution without any active tessellation sort of dynamics, right?
Okay.
And then the other question was just more basic, which is just, I think I missed it, I'm sorry, was it phosphorylation of ERK that was the signal that's exchanged to drive the wave,
Yes,
the traveling
yes,
wave?
yes,
or
yes, yes.
it was, and that's super
Yes, yes.
fast,
Okay,
is that
very
right?
good,
Okay,
yeah.
okay.
Okay. So what can be the null model in tessellation? So the null model is that you start with some seeds, okay? So every cell, and let's say you make a Voronoi tessellation of the system. And from then on, you can be working with this Voronoi cell as the domain. You half it and then you quarter it, okay? So this is the scenario on the top, okay? Another version is that there is mixing. And a third version that these domains are also deforming. So the simplest tessellation is the breaking up of a chocolate bar, and we know that this is not the case. So already in a system,
But if you add a little bit of, you know, motion, like if you motion, you will get this.
Yeah,
yes?
absolutely.
That would be
Yeah,
the
so-
generic.
yeah,
The generic,
yeah, yes.
so one would, okay, so you're not saying that the tessellation is an active process.
No.
It could very well be a passive, like.
It is active in the sense that it
Yeah.
is a consequence of activity of cell cycle, which is very much active. But
Yeah.
we don't think that tessellation then feeds back and influences the process of tessellation itself.
Right, and nor do you think that there's a mechanism that's making the solution be a tessellating solution as opposed to,
No.
right? It's
No.
just like the chocolate
It's
bar
the
plus
fact that
some
they're
noise,
sitting in the common
some jitter.
cytoplasm. Yeah.
Yeah,
It
yeah.
just, I'm a big fan of Escher, and
Okay, and
it
the
was
synchrony-
irresistible analogy to
Right,
me. yeah.
and the synchrony is essential for the Drosophila because of the rapid time
Okay, so
scale
the
of the
synchrony-
cycles?
okay,
Like, why is
the-
it so important?
Okay, the synchrony is important because you are deposited in the environment, and you want to go through development as quickly as possible before the bird comes and eats you like a larva. Okay, but by the time you become a hatched larva, a lot of things are happening. A whole day will happen even in Drosophila. So what is important here is that you establish spatially uniform nuclear density. Because once you have a spatially uniform nuclear density, you'll have uniform density in an epithelium once it cellularizes. And once you have a nuclear density in the epithelium, you can start going into gastrulation. Because if you start bending this, so you probably know from the work of Adam Martin here that very much like you form folds during the formation of the neural tube, you form the mesoderm, okay? So in order to have a symmetrically placed ventral furrow, you need to have spatially uniform nuclear densities. Otherwise, you will have an insect version of scoliosis.
Right, yes.
Okay. Yeah. Okay.
Okay, we have time for at least one more question.
Oh, hi. Thank you for the talk. I was curious to ask about the synchrony because I guess there's like the pole cells at the end that seem to orchestrate the synchrony, but there's
It's not the pole cells. It's something at the poles. Pole
Okay, so
cells
there's
also happen to be at the pole, but they get out of the embryo. But there is modification of the extracellular matrix that activates the ERK signaling pathway only at the poles.
But there's some pacemaker that's orchestrating.
Yeah. And you want to ask, where does this come from?
I guess more so that there's there's examples of synchronizing oscillators like fireflies or even just like the Kuramoto model that synchronize without like
Absolutely.
a global signal.
Absolutely. But it depends on the nature of coupling. So for example, the Kuramoto model is, the vanilla version is an all-to-all coupling. So if you start making coupling local, as you have, because you have diffusing coupling in the syncytium, the system can break symmetry, and you saw that it does.
So in this case, the synchrony dissolves because the coupling is not strong enough.
The coupling is not of the Kuramoto flavor.
I see. And do you think, or I guess what exactly are like the theoretical implications of like adding the oscillators to the system?
Oh, because you start your life as one cell. And you were also one cell at some point, right?
Yes.
And now you are much more than one cell. So adding oscillators to the system adds cells.
I guess in terms of like the synchronization dynamics, I guess is there
The
like
synchronization is
impact?
important because you want to do something quickly, and you want to do so such that the end product at the beginning of the third hour of development is spatially uniform nuclear density that allows you to then convert a two-dimensional field of cells into three-dimensional organs when you start doing gastrulation. And for this, you need uniform density, and for uniform density, you need synchronous divisions, and for synchronous divisions, you need synchrony.
I see. Thank you.
Okay, so unfortunately, we're out of time.
Well,
Or
you just
one more
have you have
question?
to close with something for mammals. So there's no evidence of synchronization in esters pre-implantation.
There is definitely synchrony up until 8 cycles of development. And then something magical is happening such that you push some cells inside and they start forming the inner cell mass. So the reason why the system breaks, so there is a natural symmetry breaking event, which is the first cell fate choice in a mammal that sends you either towards the future inner cell mass or to placenta. So in that system, it emerges spontaneously. And it happens to happen at an 8-cell stage. And this is something that is happening at an 8-cell stage in all mammals. So this is very interesting and still
8 cells or 8 divisions?
8 cells.
Oh.
Yeah, so when you're when you are a 8-cell
divisions,
mammal,
3 divisions.
you have 8 cells and you look like an orange, where every one of the cells
Look at the molar state.
Exactly. So every one of the cells has access to the surface. And then when you go to 16 cells, there is absolutely no way to fit them all on the surface, and a small group of cells, like 2 or 3, end up inside. And this is a purely self-organized symmetry breaking event. So there is synchronization, it just lasts much shorter period of time. Okay.
Okay, so let's thank our speaker again.